So welcome everyone. We are have just opened the broadcast, and so it's going to take a minute for people to be able to join us. So
Hi there.
good morning, Sudeshna.
How are you?
Excellent. So I think for this first part, we'll just have the, for the business meeting, we'll just have the cameras for myself, Yorgos, and Sudeshna. And everyone else can be off video, and then we'll be asking our presenters when their presentations come up to turn their cameras on. So Alden, if you can shut off your video. And so our numbers are stabilizing pretty good. So it's 10:03. So why don't we start so we have plenty of time for our presentations. So welcome everyone to the NACC Fall meeting. This is the Data Core Leaders meeting. And we, after 20 years, have moved from our anchor position to being the lead core presentation for this virtual year. And we really appreciate that our West Coast colleagues are getting up at well before 7 a.m. for this meeting for them. And for those of us on the East Coast, it's a little more comfortable. So thank you very much for joining us. And we're going to just move into our business meeting. If I can somehow make the slides go forward. So first of all, I'd like to thank Mark Espelon, who has been part of our Data Core Steering Committee for the past 3 years. All I can say is Mark is such a kind and skillful leader. The very first time I met Mark at Wake Forest University, I was impressed by his intelligence, his generosity, and really his sincerity and kindness. And every single time I've had an opportunity to work with Mark, I maintain the greatest feelings of just how impressive he is and how much he has promoted aging research and biostatistics and the many efforts he led, including starting our webinar series. So we have all benefited from Mark and his leadership with the data course leaders, and he will, of course, continue on at Wake Forest. So thank you very much, Mark. And so now I'd like to, as your outgoing chair, I'd like to welcome Yorgos Tripodis, your incoming chair, and we'll have him say a few words.
Hello all, thank you, Heather. I've, big steps to follow from your leadership. We'll continue the good work, and especially as we'll discuss following the ADRC panel recommendations. I think we have a lot of things to discuss on our strategy as data course. So thank you all. If you have any suggestions, questions, please email me at yorgos@bu.edu, and I can reply as soon as possible. Thank you.
Great. I'd also like to welcome Sudeshna Das, who is our chair-elect, and I'm going to have her say a few words.
Thanks, Heather. I'm really looking forward to and very excited to for this opportunity. I'm a computational biologist by training, and my particular research interest is in integrating various kinds of data modalities, everything from molecular omics data to clinical phenotypes. And I hope to be able to break down barriers that exist between these different data types, sometimes between institutions and sometimes within institutions. So that's what I hope to do. And I also have a deep background in informatics and building software. I've been in the biotech industry for 10 years, so I hope to bring that to NACC and the several cores. And I'm particularly excited that this time we are the first session instead of the last, as Heather mentioned. And it's been my pet peeve that when the data session happens, pretty much most of the audience is gone, at least for the past couple of meetings that I've attended. So I'm very excited that this year we have a lineup that is hopefully has broad interests, and hope to continue in the footsteps that Heather and Yorgos and Mark have set. Thank you.
Great. One of the things that I think we're really trying to do is show how the data cores are integrated. And so we'll be having leaders from both the neuropathology cores and a rec core as our panelists, and I'll touch on that shortly. So we'd like to have a little check-in from our other data core members, just to kind of give you an idea how we're trying to flow this virtual meeting. Is that we will be having some poll questions during the meeting, so that you can be giving feedback. These will be pop-up things on your screen. I'm not exactly sure how they're going to happen, but we're trying to do that. And so we're trying to figure out a way to allow some feedback about how things are going at your ADRC during this COVID time. We know that we've been having to develop new forms for televisits. We just had the new information sent out about form changes for the neuropath forms for RTAG. There's also optional forms that have been being sent out to be created. So we're just going to kind of open it for a second to see if there's any feedback from the audience. And I realize in the format of my screen, I can only see slides and I can't see the chat. So I'm going to rely on Yorgos and Sudeshna to allow us to get any feedback. So I see someone raise their hand. Can we unmute them?
Yeah, I'm looking for the hand.
I saw a hand go up. Ellen Weissman raised.
Oh, got it.
And I think there was another as well.
Okay, Ellen is unmuted.
Hi, can you hear me?
Yes.
Here on the West Coast. I've got my brain cells moving forward. So, you know, we're scrambling, but we're actually doing reasonably well, I think. We've got, we were not doing electronic data entry, and we've been we were moving towards that this particular round of funding anyway. But we've been scrambling, and we seem to be, you know, getting there. It wasn't exactly what we were intending to do first this year, but we just have rethought our priorities and what we're doing when. And so some of the things we've done is we do not prioritize speed of getting stuff through, although things are going through pretty fast. But we've been prioritizing getting the most important things coded up for our clinical core so that they can do the data entry sort of virtually with their virtual visits. So, you know, it's moving along. I would say, you know, it's not perfect, but we're doing what we can. And, you know, it's just taking a lot of communication between our data manager and our clinical core people. Say, what's the most important thing to do? You know, what would help you keep track of where you are when you're distributing all of this data entry among your various people who are collecting the data and so on. So it's going okay. I wouldn't say it's wonderful, but I think we're doing okay. I don't know how other people are doing, but I think it's okay. You know, we still haven't gotten everything cut, you know, the new forms. They're not all done yet, but you know, we sort of sometimes we've been in this situation, and other times when we had a backlog of something, we sort of would say, okay, we can fill out these forms later or something. So you know, we can fill out the most important stuff and get to some of those other things later, and then do them all in a big batch or something. So that works. So I don't think it's horrible. It's more that, you know, we've been trying to think about what's most important to keep going. And I think, you know, keeping the clinical visits such as they are, you know, moving forward so that they happen is important for this.
So your center's able to have in-person visits?
No, not in person.
Okay,
It's
they're virtual. Okay.
It's all virtual visits, right? We've had a lot of telephone visits. We're an old center, and so we have a lot of old people. So we have plenty of opportunity, plenty of experience with tele visits for people who can't come in as it is. But now we're doing everybody that way, and it's working. You know, it's just there's some things we can't do, and that's the way it is.
Great. Are you using the supplemental form on the access to technology?
That's some of the stuff we're working up, the new forms. You know, we're trying to get stuff coded so that people can get those things filled out. I don't know, well, maybe on the call, but I don't know if that's actually done yet. It's on the list of things to get done.
I know for the Yale data core, it has been tricky to keep for everything we're doing, for our amounts of effort to code up all the new forms, be doing everything remotely because unless you have patient care or other certain responsibilities where you must be on campus, we're all remote. So everything is a little slower because we're like connecting through all these firewalls. And then if something breaks in that firewall chain, it can be quite time consuming.
Yeah, we have the same general issue as well, because it is slower to do some of these things. So there are a couple of steps that are really slowed down by being remote. We have I guess it's really during the summer, the middle of the summer, there's been some relaxation in terms of our ability to go in. So we coordinate within the group so that no more than one person is in our space at once. So we have a coordination going on, and we have other projects too. And so we coordinate within the group to make sure that there's no more than one person at the office at once. In which case, you know, like Will, who's the data manager and has to sort of push the stuff up to NACC and everything, you know, he needs roughly maybe a day a week that he's in the office to do some of those things because they're a lot faster. And that's been helpful.
So I would like to just let people know in the
Yeah.
question and answer area, we're starting to populate that, Susan Hunt shared with us that the REDCap data dictionaries for 2 of the COVID forms and telehealth forms have been built and are shareable. and Sudeshna would like to make a comment on that.
Mhm.
And also Nina Silverberg has written in, and I will, Yorgos would like to respond to that. So
Sure.
folks, keep looking at the question and answer, but first let's hear from Sudeshna.
Next person. Mhm.
So thanks to Suzanne and the rest of the team. Those forms are available. So Ellen, at our center, just like yours, we weren't doing remote data. We weren't doing electronic data capture. We had a project going on to transfer to REDCap, and everything kind of had to be like accelerated because of COVID. So did I hear you? But if the COVID surveys are out there, both the technology and so the REDCap forms have been built. I mean, deploying it is still challenging, but at least those forms do exist. And then once the other issue, I don't know whether other centers have been facing, we've gotten about like 65% response, and those came pretty fast, but the rest of the participants has been a little challenging to reach out to make sure we get like a higher participation. So wondering what other people have to say. But I'm going to let, there are a few other questions respond to, people have a chance to respond to those first.
Yorgos, would you like to have your response?
It is not really a response. I was
Okay.
just wanted to emphasize what Nina said, and tomorrow we'll have some presentation of the results in the director meeting. So I think data core leaders should really participate.
Great. I want to keep us on time, so we only have 2 more minutes before the presentation starts. So I'm just going to touch upon some of our past year's achievements based on the ADC panel recommendations. So we really have been trying to leverage existing data and computer systems. Largely, we've been doing this through the webinars and our previous analytic workshop, which you'll be getting a poll question as to whether you would like to be having these in the future. And just as we had the sharing of the new REDCap forms for 2 of the COVID forms, we're encouraging sharing of REDCap and other systems codes as we have been doing. So I think what we probably need to be doing is thinking of some sort of format so that's centralized, so data cores can see what's out there and how that fits their particular systems. For the upcoming year, we'll have Yorgos say a word, and then I will give an introduction to our presentations. Yorgos?
Just a quick word so we are on schedule. As I said in my introduction, we have the panel recommendations, and that will form our strategy. And based on that, we'll also would like to continue the webinar series and possibly expand them on methods as well as data sharing. So please email any of us, Heather, Sudeshna, or myself, for ideas for upcoming webinars.
Excellent. So we're breaking our presentations into 2 parts so that we have 2 presentations and then a panel discussion. So please be very active. You can be typing your questions into the question function throughout the presentations, and we will, the panelists will be asking questions, and we will also be trying to integrate your questions in. If there is not time, then the speakers can be typing in and answering your questions. So in the first 2 talks, these are the first one is going to be the Alzheimer's Discovery Portal, which is an informatics tool for addressing the translation gap. And that's going to be presented by Robert McDougal, who's a new assistant professor at the Yale ADRC and our data core, and Professor Caroline Zeiss, who is a double-boarded pathologist and is also our the Yale's neuropath co-leader. And so this is these two talks are going to be from people with a bioinformatics and a translational view. The second talk will be regression techniques for right-skewed data in preclinical Alzheimer's disease by Malek-Ahmadi from Banner's Alzheimer's Institute. And Mike is an informatics scientist, and so we're looking forward to his talk. And we're really fortunate. What we're really trying to do is bring together other cores to the data core meeting. So we're excited to have Peter Nelson from the University of Kentucky. It's ADRC. He's their neuropath core leader, as well as a leader of the neuropath core's steering committee, and Sudeshna Das, who is our chair-elect and has a informatics background. So I will introduce the next two talks after our panelists. So I am going to stop my share, and Robert, if you could share your slides, and I will go on mute.
Hi, everybody. Can you all hear me?
Yes.
Great. Thank you for that introduction, Heather, and welcome to everybody meeting, especially those folks on the West Coast. I know it's early there. So my name's Caroline Zeiss. I'm going to be discussing the first part of this talk, and then we'll hand that over to Robert. We'd like to describe a program we're developing to support translation of discoveries in animal research to the Alzheimer's patient. The first version of the program was developed in my lab in collaboration with Halil Kilikoglu at the NIH. He is now at Illinois. Under Robert's care, we're now expanding this with Halil's continued collaboration. Next slide, please. The translational gap refers to the phenomenon that most results in animal studies do not translate to the clinic. This is a general phenomenon, but it is particularly profound in AD. Next slide, please. The reasons for the translational gap are complex. Poor reproducibility is one frequently cited reason. This refers to the failure of one lab to reproduce results using the same approach as another lab, and it concerns variables that are central to the actual conduct of the experiment. The NIH has developed a database called ALZHEIME'S that explores reproducibility in Alzheimer's disease. Our intent and our interest is really much more in translatability, which refers to the ability of results from animal studies to generalize to human populations. The variables affecting translatability are much more contextual. In particular regard to Alzheimer's, a challenge is that most available models are reductionist in nature and attempt to model a disease that is very complex and has significant mechanistic and clinical overlap with related dementias. So with that in mind, we spent a couple of years manually mining the literature to identify those animal study design variables that associate with successful translation in AD. We also included Parkinson's disease, where we have more examples of successful translation for its motor symptoms. That knowledge supported the design of Menagerie, a text mining program, which is designed to collect data from PubMed abstracts in real time. Menagerie extracts data to populate a database composed of the modules depicted in the colored boxes. One unique aspect of the program is that it identifies whether a functional outcome measure was used to assess efficacy of an intervention in animals. We felt this was an essential variable to report if results were to be translated to patients. The program is written in Java and combines rule-based algorithms, signal lists, and machine learning. It incorporates some existing tools such as SEMREP and GNOME+, and uses resources such as the Meta Thesaurus or MESH to support entity recognition. Next slide, please. Next, we extend a menagerie to interrogate the entire Parkinson's disease corpus. That database, and this is at the abstract level, that database is publicly available at the red link below, with some examples of the patterns one can extract. Just a note, could you go back, Robert? Just a note, in the top graph, the proportion of positive or promising studies in humans is that high because most studies that occur in clinical trials do not make it into publication, and the ones that do are biased towards those that are successful. Okay, now we can have the next slide. One can use the data to perform various experiments. For example, here in this experiment, we mine data from 44 Parkinson's disease interventions, some of which were FDA approved and some of which were not, to see if we could determine patterns of animal use that associate with FDA approval. Next slide, please. We generated a large number of hypotheses, such as the example shown here, and tested these using logistic regression. From this, we determined an overall pattern of species, model, and functional outcome measure choices that's significantly associated with FDA approval. Next slide. Those that were significant are summarized here in this visualization, with species and model choices on the y-axis and use of functional outcome measures on the x-axis. The size of the bubble indicates the number of studies for that intervention. And for clarity, only a few interventions are labeled. Using this kind of visualization, you can very quickly see that transcranial magnetic stimulation, although not FDA approved for Parkinson's, shares many characteristics with approved treatments. This approach allows one to explore PubMed data in real time to assess the translational potential of any intervention that is reported in there by determining where it exists within this matrix. And so our job now is to extend this to Alzheimer's disease. And with that, I'll hand you over to Robert.
So building on this framework, we're developing Menagerie 2.0, which will characterize the Alzheimer's literature with the ultimate goal of identifying gaps in the literature with the greatest potential for providing evidence for or against an intervention's translational potential. From the biomedical side, we're introducing modules for detecting new categories of metadata about a paper's topics, identifying new relations, and new types of animal models. Professor Zeiss and her group are generating training and test data for these modules by annotating papers using BRAT. From the data processing side, the main changes are that Menagerie 2.0 is designed to quickly incorporate updates from PubMed and to place greater emphasis on interactive online data exploration and visualization. This process is enabled by a number of technologies and resources. I'll highlight a few. Ontologies, formalized representations of, in our case, biomedical knowledge, with terms, identifiers, and relationships are essential for this type of literature analysis. Fortunately, there's a number of good ones out there, many of which are freely available, like MESH, that we know from PubMed, which can tell us that any of a number of terms that we might use to describe Alzheimer's all refer to the same concept, which is both a dementia and a tauopathy. MESH tells us about known pharmacological actions of chemicals, which can help us infer why they're mentioned in a publication. Our characterization approach builds on MESH, builds on other ontologies, including the UMLS, a metathesaurus of many ontologies, with a relatively restrictive but still free license, and an extensive ecosystem of tools, several, like SIMREP, developed by our co-author, Professor Kilkuglu. While ontologies are essential for identifying terms and semantic relations, they don't necessarily capture the context or have the full specificity we might like. For example, here's PubTator's ontology-based annotations on the abstract of a 2017 paper. Disease-related concepts are in orange, species-related terms are in light blue, genes are in purple. It correctly identifies the two species, mice and humans, are referred to by this abstract. But if you look closely, you'll see that humans only come up in the introduction and in the conclusions. They're just there to frame the research, which was done in 3xCG mice. Here, the genericness of PubTator means that it doesn't annotate the specific type of mouse model. But that's essential to us since we want to be able to characterize the Alzheimer's literature specifically. A set of rules have been developed and are being refined to transform automated ontology-based annotations into what we need to characterize the Alzheimer's literature. We don't have the numbers for Alzheimer's just yet, as we're still annotating papers, but this general approach has been validated with Parkinson's, where we're shown to improve both precision and recall. The rules vary depending on the category, but most of the time they're actually fairly straightforward. For example, we have a list of known Alzheimer's-specific model organisms that we cannot afford to miss, and we intend to enable semi-automatic updates of this list by identifying patterns of word use. We have exclusion criteria. We've already seen, for example, that species in the introduction of an abstract do not necessarily mean that the paper is studying those species, and we have ways for inferring the introduction when it's not a structured abstract. Some usage patterns and forms indicate things that could be genes might not actually be genes, and so forth. Positive and negative outcomes are inferred by two separate but closely related machine learning algorithms using features extracted from the text by the bioscores package. Obviously, this analysis generates a fair amount of data. A highly abridged snippet from one paper is shown here. Some of the main considerations are that the data ends up being hierarchical, and that multi-valued fields and missing fields are common. For example, one paper might measure many functional outcomes, but not explore the genetic basis at all. This structure is more naturally represented in a document-oriented database rather than in a traditional database. And so we're storing both the derived and the original data in the document-oriented database MongoDB. We store the original data along with the derived data to give the most options for data exploration. Fundamentally, the goal, after all, is not to derive data and have it sit there, but rather to make a tool that you, the community, can use to help you identify paths to doing research with the greatest impact. As such, we're trying to make a tool that gives you the most options right on the tool's webpage. No downloading a big data file, no needing to learn our schema, no writing custom code. Something that gives you the most options for searching the data. Here, for example, on the right, I did a full text search for all the blank mouse models. We see TG2576, PS1, and 5XFAD are all towards the top of the list. We want you to be able to visualize trends in coverage for cross-referencing data to find out, for example, what genes get studied with what model, how many distinct authors have explored a given model intervention combination, etc. By the way, if you have any suggestions as to types of analyses you might want to do, feel free to contact us and let us know. Obviously, while we want you to be able to do as much as possible on the website, you may reach a point where you need to download queries, and we're trying to make that as painless as possible with multiple formats. Here's a screenshot from the prototype search result page showing how references in the Alzheimer's literature to APOE, a protein implicated in Alzheimer's, ramped up rapidly from the early to mid-90s and have increased relatively slowly since then. Of course, we get a list of papers that we can click on for more information. We also see that this has been studied in 17 classes of animals or animal-like systems, at least based on the titles, and that's been looked at by almost 13,000 authors, and we can see who's taken the lead on that. We intend to add more graphs like the ones Caroline showed earlier, with the trends on different categories like functional outcomes over time for a given query. The trick is to make all of this easily discoverable without becoming overwhelming. This example is a single text query for simplicity, but we're expanding support for ontology-based queries and have a version that allows comparing trends across multiple queries. So you could compare, for example, the measurement of functional outcomes of 3xCG versus 5xFAD mouse models. I want to turn very briefly to two future directions. First, we're very interested in fair data sharing, which is a modern effort to formalize on a conceptual level what's needed for computers to work with data and to combine data from other resources. In brief, fair data must be findable. Amongst other things, it needs to be a permanent identifier. It must be accessible using standard methods, even if requiring authentication. More on this in a moment. It must be interoperable, which in part means using standard ontologies, and it must be reusable. Ontologies come in here, but so does licensing. As far as interoperability goes, we'd like our stuff to use compatible terminology even when we venture outside of the formal ontology space. So I'm thinking, for example, of exploring ways to work with ALZHEIME'S list of Alzheimer's models and model types. There are also some licensing issues, questions about what types of capabilities for automated data exploration are most beneficial to the Alzheimer's community. You'll notice that everything we've talked about so far has been with respect to information derived from abstracts. This is because they have a relatively high information density and because they're relatively freely available. That said, we're fortunate that a relatively large percentage of Alzheimer's papers in PubMed Central have a permissive CC BY license. And so the second future direction I want to highlight is that in principle, this allows us to derive data from their full text, which we can get in a standardized structured form, allowing us to focus on high information sections like figure captions as illustrated here. I'd like to thank the many people that have played key roles on this and the funders that have made it. Thank you.
Thank you very much. And we will be, I can see there's some questions in the Q&A section, so you can be addressing those while we hear our next talk from Mike Malek-Ahmadi. So I'm going to hand the screen over to you, Mike.
All right, thank you, Heather. I appreciate the opportunity to speak in this forum and represent the Arizona ADC. So my talk today is going to focus on, pardon me, my talk today is going to be focusing on regression techniques that we can use with RIGHT-SKEWED data, in particular when we see this kind of data with preclinical Alzheimer's disease applications. To begin with, I just have one financial disclosure I want to point out. And I also want to acknowledge my many mentors and colleagues who have worked on the various aspects of this part of the work you'll see here. In particular, several of them worked with me on papers that were generated from the analyses and examples that you'll see in this presentation today. So we know, generally speaking, that linear regression, it assumes that the error of an outcome variable follows the normal distribution, or bell curve, if you will. However, we all know that there are many cases where the data are heavily skewed, particularly toward the lower values for a variable. And this is really relevant for preclinical Alzheimer's disease, where lower values on a particular scale or measurement are likely to have higher frequencies. So we can think of, things like, amyloid imaging measures and other biomarkers that are likely to have this sort of, skewed distribution. So an example of what I'm referring to, if we look at some neuropathology data, we look at neurofibrillary tangles, neuritic plaques, and diffuse plaques, and we're looking at just, counts, from different cortical regions. We can see that we have this very highly skewed distribution toward the lower values of these particular variables. So the way that, skewed data is often addressed, the first and most common way is probably using transformations, in particular log transformations. But one problem that log transformations have is they do not necessarily yield normal distributions. And you still have to perform a normality test on your transform data to ensure that the log transformation actually did what it was supposed to do. I know that several papers that I've seen and actually reviewed myself, many of the investigators don't actually do this. So this is one thing to keep in mind, that the transformation itself may not fully transform the data in the intended way. The other sort of sticking point with using log transformations is that the interpretations of the transform values can be difficult, and you need to be able to interpret the values on their original scale. There are ways to do that which are beyond the scope of what I'll be talking about, but I just want to bring this up as one, sort of roadblock that comes with using log transformations. Another, approach that is often taken is dichotomization, where we simply dichotomize a continuous variable at sometimes an arbitrary point. The downsides of this are a loss of statistical power. The clinical and scientific meaningfulness of the cut point is often, questionable. However, there may be a few instances where dichotomization, may be valid, and that's if you're using a clinically or a scientifically valid cut point, such as an HbA1c value, an AV45 SUVR value, and such like that. But generally, when we see dichotomization happening and being utilized, it's often at some arbitrary value, such as the mean or median or something else. So one of the first major approaches regression-wise that can be used in this kind of right-skewed data is Poisson regression. And it's often used to characterize count data where the lower values of a variable have the highest frequency. And some examples where this has been utilized is in the number of arrests and number of goals scored in the World Cup, just to provide a couple of different examples. One of the major assumptions of the Poisson regression models is that the mean and the variance of the outcome variable are equal to each other. But what happens in instances where our variance is very high, as we often have in preclinical Alzheimer's disease studies? So one way to approach this and to handle this is to use negative binomial regression. So the negative binomial model is similar to the Poisson model, but what it does is that it incorporates an additional term to account for excess variance. And very much like the Poisson model, the negative binomial model can be used to characterize count data where we have the majority of data points that are clustered toward a lower value of the variable. And again, the key difference here is that the negative binomial model can be used when variance is substantially higher than the mean. So I want to walk everyone through sort of a case study in which we compared the performance of a Poisson regression model and a negative binomial regression model using some neuropathology and cognitive data. So in neuropsychology, the term dispersion refers to the degree of variation of performance between different cognitive domains for an individual. So when we're looking at an individual's performance between, say, executive function, episodic memory, and other domains, when the term dispersion is simply referring to how different that individual is performing between those different domains. And several previous studies have found that individuals with higher cognitive domain dispersion are at an increased risk for developing Alzheimer's disease. However, prior to the work that me and my colleagues did, there were no studies that linked cognitive dispersion to the neuropathological findings of Alzheimer's disease. And the paper that the example I'm showing you is based off of was published in the Journal of Alzheimer's Disease in 2017. I've provided the reference here for anybody who might be interested. So the data that we used came from the Rush Religious Order study, and it was 123 cognitively unimpaired older adults who had both cognitive and neuropathological data. And we were using 5 domains, episodic, semantic, and working memory, as well as perceptual speed and visuospatial function. And we used 3 measures of AD neuropathology, which were neurofibrillary tangles, diffuse plaques, and neuritic plaques. And each of these measures represents a summation of counts from 5 different cortical regions. And so our main research question was, does within subject cognitive domain dispersion, is it associated with AD pathology? So the primary metric that we used to address this was something called the intra-individual standard deviation. And we use this as the measure of cognitive dispersion. And this is the most common way that cognitive dispersion is quantified in other studies. And what this is, it's simply the standard deviation of the cognitive domain scores, for an individual subject. And so what we did is we used both Poisson and negative binomial regression models to assess the association between intra-subject standard deviation and the 3 measures of AD pathology while adjusting for age at death, sex, education, and E4 carrier status. So again, I just want to highlight what the distribution of our neuropathology variables look like. As you can see, they are highly skewed and definitely heavily right skewed, but certainly there is a lot of variability in terms of the range in which we see the different pathology measures. So just some basic descriptive statistics for the neuropathology measures. I'm showing you the means, the variance, and the standard deviation for each of the variables to sort of set the stage for how the regression models that we're going to run may turn out. So when we actually run both the Poisson and the negative binomial regression models, we do see some very stark differences. So one thing I want to point out in the top table, where we have the results from the Poisson models, is that the residual deviance values are quite high, and these p-values are indicating that we have statistically significant associations. Now, when we go down to the negative binomial models, we can see that the regression coefficients are very much similar to the ones that we saw in the Poisson models, but here we see the residual deviance values are much lower, and now what we're seeing are p-values that are indicating a lack of association between intra-subject standard deviation and these neuropathology measures. So our question then becomes, well, which of these models is correct? So one of the ways that we can sort of tease out and make a more definitive and empirical assessment of our model fit is by testing the residual deviance values in the chi-square distribution. So for all of these models, they have a degrees of freedom of 116, and this corresponds to an expected chi-square value of a little over 142. So what we would take from this is that residual deviance values that are lower than 142 would indicate a good model fit, while those values that are higher would indicate a lack of fit. So when we do a side-by-side comparison of the Poisson model residual deviance values and our negative binomial residual deviance values, we see a very clear comparison and very clear difference in that our negative binomial models do give us a good model fit for our data. However, the Poisson models clearly did not. So some additional ways that we can assess model fit, and this is using R for anybody who might be interested, is we can actually get a quantified p-value for the residual deviance value. So by using just this 1 minus p chi-square function within R, you can actually get a p-value, and this p-value would then again indicate whether or not you have a good model fit. An additional method for assessing model fit was proposed by Allison and colleagues. It's a little bit it's a little bit more subjective and not as clear, but basically what they suggest is that when the ratio of residual deviance to the degrees of freedom for a model is equal to or approximately 1, then you have acceptable model fits. The one thing that is lacking in this approach is that there's no sort of specified cutoff or range in which when a value gets to a certain point beyond 1, whether it's considered to be a good or bad model fit. So this last bullet point is more of a guideline than it is an actual sort of objective empirical way to assess model fit. All right, so in the examples I showed you just now were where we're dealing with integers or count data. But if we're not using count data, we still want to be able to have a model that we can use to analyze this kind of skewed distribution. And one approach that we can use is under the generalized linear model, we can specify the gamma distribution to do this. The one thing that this does require is that the dependent variable cannot have zeros or negative numbers. Right, so this is just an example of different gamma distributions that are possible, but it gives you sort of an idea of how the gamma distribution can be applied to right-skewed data. So the data I'll be using for this example came from ADNI, and it's 297 cognitively unimpaired cases. And we were specifically interested in white matter hyperintensity volume derived from FLAIR images. And our research question here was, simply, was AV45 SUVR associated with greater white matter hyperintensity volume in these cognitively unimpaired subjects. So here is the distribution of our white matter hyperintensity volume values that we derived using our normal pipeline. This was using the lesion segmentation toolbox that we see within MATLAB. And so when we have white matter hyperintensity volume, we were using that as the dependent variable with AB45 SUVR as the independent variable. Our covariates for our regression model included age, sex, education, the Hitchinski ischemic scale. We use that as sort of a measure of vascular risk, and also ApoE4 carrier status. And on the third bullet point here, I've simply put in the code that I used for the model in R that I used to run it. Okay. And so here is the result of that regression model. So here we can see that AB45 SUVR did have a significant association with white matter hyperintensity volume. And importantly, to assess our model fits, we can find our residual deviance value and the degrees of freedom down here. And from there, when we actually do when we actually test the model fit within the chi-square distribution, we get a p-value of approximately 0.06. So just under what we would consider this to be a good model fit for this particular data. So generally speaking, when data are right-skewed, we can utilize Poisson, negative binomial, and gamma regression models to use our data. What this does is it allows us to not have to utilize transformations, which can make the interpretation of our data problematic. And in addition, as I mentioned in the first part of the talk, is that even after applying log transformations, we still may not obtain a normally distributed set of data. And then, of course, these regression methods, I believe, will be especially helpful in preclinical Alzheimer's disease studies, where the distribution of our imaging and biomarker data are very likely to be right-skewed. And so that will be the conclusion of my talk, and I'll be happy to start answering questions in the Q&A section.
Now we welcome our panelists, Pete Nelson and Sudeshna, to lead the panel discussion. So we could have Caroline and Robert also turn on their videos.
Dr. Das, I'll follow your lead since you're a card carrier and an actual member of this tribe. So I do have some questions, but I'm not sure how you want to organize it.
Yeah, all right. A fascinating talk about addressing the translational question, which is kind of like the central theme of Alzheimer's disease, unfortunately. So great talk. And my question I just wanted to start off with, and my colleague, Dr. Nelson, has some additional questions. There seems to be a lot of manual effort that was used to build the test and training sets. So I was wondering if either of you could give us a sense for what is involved in creating such large test and training sets, and the importance of having like a good one and not a biased test, and your strategies to select those.
Sure, I can answer that. So yes, the answer is that it's an incredible amount of work to develop those annotation sets. We use a program called BRED, which makes it a lot easier. And some things are very easy to annotate. Entities are quite easy to annotate. But contextual relationships are much harder to annotate. So what we end up doing is going through several rounds of annotation where we have several different people annotating, and then we have that reviewed by another person. We also define certain rules to sort of limit how we annotate things contextually. And we stick to those, we revise them occasionally, but at the end, when we have the final annotation set, we go through everything and make sure that we've stuck to those rules, because they tend to change over time as you annotate.
And so what kind of effort do you have, like full-time people doing this curation and annotation, or
No, we have undergraduate students who don't know what they're getting themselves into.
Yeah.
And we have a couple of faculty members, but everybody's doing it when they have time. And luckily, it's the kind of thing that you can sort of do in between other things.
Cool. Yes, unsuspecting students have been our strategy for building tests as well. Yes. Dr. Nelson, you have some other questions?
I guess the Q&A also had some questions related to data sharing and whether or not you could get to an open sharing spot like GitHub, and how the nomenclature can confound things. Did you want to discuss that, Dr. McDougal and Dr. Zeiss?
I can
Robert, do you want to handle GitHub?
Yeah, I'll handle GitHub, and you can handle the nomenclature.
Sure.
All right. So as far as the GitHub goes, the actually I should have let Dr. Zeiss do this. So we're building upon previous work, as mentioned. So the Parkinson's work, which does a similar analysis, the code is available on GitHub. I did put the link in the Q&A part, so you can go ahead and check that out. So that's calibrated not to Alzheimer's, but it at least gives the core code, which is in Java, and there's a paper that discusses it. Everything else is being, so the discovery tool that I demoed, well, showed screenshots of, is available. It's being developed on GitHub, but it's not publicly released, but we will eventually do that as well.
Okay, great. And then really nice talks. It's a pleasure to be here as a neuropathologist. I appreciate you all having me here. I'll go then to Dr. Malek-Ahmadi for a quick question here. You mentioned sort of the difference between dichotomization, which I think has caused a lot of problems in our field. Saying that you are AD, yes, no, you're demented, yes, no, is a huge problem that has caused all sorts of confusion, whereas you have much more quantitative metrics, which I think are a great strength, but it's usual that you can't do that for big studies that use multi-centers. What they use for the neuropath is usually semi-quantitative. Could you remark upon whether or not some of your models could work? And again, these are things that we see all the time, these problems of over dispersion and zero inflated counts with the models, very troubling, but how that they work with semi-quantitative metrics of the neuropathology and the clinical pathological correlation.
Yeah, I thought about that because I know that when we think of something like the UDS data set or even others that other neuropath data sets, they don't really give us that granularity of the actual counts for that. It does start to pose a problem because you do have sort of your distribution being condensed into sort of these not necessarily categorical bins, but you basically are restricting your range. So I haven't really been able to think about and put together a reasonable approach for those, other than using categorical analysis methods such as ordinal logistic regression, multinomial logistic regression for those cases. So again, it doesn't sort of really address the problem of this dichotomization and sort of putting continuous variables into categorical ones, but it's as for right now, those are the approaches that I've taken.
Good points. And then before going back to Dr. Das for another round, let me ask Dr. Zeiss and Dr. McDougal again, something that I was thinking about. It sort of depends on who your audience is for your web application. It's a really important question for me. And when people think of translation, they think of from the bench to the bedside. But I think for informatics, it has to go in two directions, both from the bench to the bedside, but also from the clinical information back to the animal modelers. And when I think of the difference, particularly in Alzheimer's disease, more so than Parkinson's, the importance of the complexity of the cerebral multimorbidity that occurs in aged people that underlies the AD dementia complex is very, very challenging to model in these smaller species, and how do you get that information about the complexity to be expected in the human, ultimate animal model, to convey that to the people that are modeling in the first place? And so whoever, Dr. Zeiss or Dr. McDougal, wishes to answer that, like getting that two-way street of translational information flow.
Sure, I can answer that. So in this newest iteration that we're developing, we have a neuropathology module. We also have an imaging module. We have a biomarker module. And we annotate pathology lesions. And so anything, for example, a vascular comorbidity in people would be annotated. And it would associate with the human species. You're absolutely right that animal models do not have those comorbidities, especially vascular. I think that's one of the real drawbacks. What you're going to end up with when you do searches for these is that you're going to have the lesions that associate with animals associate with those papers, and the lesions that associate with people associate with those. The audience are both people who do animal research and clinicians. And the intent is for somebody, for example, proposing an animal study to see what has already been done, what models have been used, what have the outcomes been, or for somebody trying to develop a drug, for example, trying to make this decision in a drug company, to see what is the full spectrum of outcomes across animal models, and if any of that has actually been translated to people.
Dr. Das?
Yeah, great. There are some questions in the Q&A, and if you could address those. So one's from Hui Zhang, who says, in highly demanding, we could develop an online convenient tool to examine data first and suggest which one of these models, Poisson negative binomial, or maybe a quasi-likelihood Poisson is most appropriate. So are there like plans, Dr. Malek-Ahmadi, to build some kind of an online convenience tools, given that R is fairly easy to do, I guess? Yeah, anyway.
No, I actually had not thought of that, and it's actually a very nice suggestion. So I appreciate the comment there. That's not something I'd really considered, but certainly I think would be very feasible, kind of given some of the capabilities that we do have, particularly with R. So yeah, I think it could be done.
Oh, fantastic. And related question, is there, there are also some zero inflated gamma models, and if you could comment on that, and as well as hurdle models. This is a question from Carolyn Zhu.
Yeah, so definitely, there are zero inflated versions of the Poisson and negative binomial and gamma models. This is when you have, very high occurrence of, values with zero. In the case of the data that I presented that I've been working with, that is not something that I come across, and I don't have haven't had to deal with. But certainly, if you do have count data that where you have a very high number of zeros occurring in your data set, certainly the zero inflated versions of these models would be appropriate as well.
Great. I have a question about your gamma distribution, and I know you talked a little bit about it, but how do you adjust the overfitting other than looking at the residual deviance? Because you're selecting yet another parameter of the distribution. So are there strategies beyond like looking at the residual deviance to address overfitting of these models?
So I've tried to think about this too and look into it. So unfortunately, within R, with the package that's used to do the gamma distribution, you don't get that nice, R squared value that we all know and love for a model. The other approach is to use the AIC values. So if you are, iteratively, building models with different covariates, you can, use the AIC values to compare, which one is giving a better model fit or not. But as far as assessing the appropriateness of the actual, model, functions to the data, the only things that I've been able to find so far that give you, a very sort of objective way to assess that is by, testing the residual deviance in the chi-square distribution. Outside of that, I have not come across anything else that would be, empirically based to do that.
Could just sort of on that note, could you discuss about using standard deviation, robust standard errors versus the over dispersion? Like, could you discuss that whether or not one is better than the other?
I don't know for certain in that particular case, but I do know that, one other approach that would be available would be to use robust regression techniques, which, do, minimize a lot of the error and do produce lower standard error estimates for coefficients. So it's an alternative approach, that, could feasibly be used as well. With the robust regression methods, those are, less common and a little bit less, well known. So it would take a little bit more introduction and, explanation on the part of the investigators to sort of talk about those and how they would be applied to their data. But certainly I have begun to sort of look at robust regression as another alternative method that can be used with skewed data.
And continuing on this, because I'm a data scientist, I think of like bootstrapping, and so the other statisticians, Jorgos or Heather, if you have thoughts on, as well as Michael, whether you could use bootstrapping to estimate the fitting and the appropriateness of the functional form of the models themselves.
This is Heather. I think one thing we should think about is really what is the interpretation, right? Mm-hmm. So I think one of the things Pete brought up is there's a continuum of the cognitive spectrum and a continuum of disease, and it's not really just this dichotomy. So you could be looking at something if you have these two-part models where you're modeling kind of the zero part and the non-zero part, and there's several parts of that. You could be like, this person is asymptomatic, they have no biomarkers or measures that show any pathology, and these are the characteristics associated with that. And then the non-zero part being, these individuals have one or more symptoms, or they have, their biomarkers are elevated or lowered, whichever is bad, to a certain degree. And among those people that would then represent a spectrum of potentially having the disease process, then we could be looking at what are the characteristics associated with that. So I think that indeed statistics has been a busy and growing field, and we have lots and lots of models to do things, but I always try and come back to the investigator and say, what is your question? And then match that technique that actually answers that question. And then when we don't have a technique that can answer that question, that points to us, what can we do to try and develop new methods to better answer questions that exist. So that does bring us to 11:10. And so thank you all. And I am going to introduce our next set of talks. Thank you. And I am just going to bring up a slide for a second so everyone sees our upcoming talks. So we're going to, our next two talks, first we're going to hear about inverse probability of autopsy weighting, moving towards best practices by Erin Abner, also from University of Kentucky ADRC, and she's a co-leader of that core. And Erin is an associate professor of epidemiology at University of Kentucky and has been with that data core since 2006 and is the associate leader. And then we're going to hear about MCI classification, so kind of really feeding right into Peter's point about the continuum. So we're going to hear about MCI classification in the longitudinal aging study in India, diagnostic assessment of dementia, the LASI-DAD, by Alden Gross, who is an associate professor also in epidemiology at the Johns Hopkins Bloomberg School of Public Health, and is a member of their data core. Our panelists are going to be Mark Bondi, who is the rec leader from UCSD and has done quite a bit in MCI classification, and Yorgos Tripodis, our new chair of the data core. So I will stop the share, and Erin, you should be able to upload your slides.
Okay, thank you, Heather. My slides should be up.
Yep.
Okay. And we are now in slideshow. Okay, so thank you so much to the steering committee for the invitation to be here. I'm so happy to be here at the fall meeting today. And this is work that I have been working on with one of my PhD students, Taylor Estep. She was a big help in the lit review portion of this so far. So inverse probability of autopsy weighting, not widespread in our field yet, but it might be eventually and probably should be. So let's talk about what this is. So I'm going to introduce the counterfactual theory of causal inference. I'm going to introduce inverse probability of treatment weights, inverse probability of censoring weights, review inverse probability of autopsy weighting as it has been done, and make recommendations for best practices. So let's start with thinking about counterfactual theory and an intuitive definition of causes. So this is what I use when I teach this to my students. So let's say Alice in Wonderland is happening and Alice is there and she finds a mysterious bottle labeled "Drink Me." So of course she drinks this, right, as you would, and she shrinks, as we know, into tiny Alice. So the causal question is, had Alice not consumed the substance, all other things being equal, she would not be tiny. Did the Drink Me substance have a causal effect on Alice's size? And normally I give people a little bit to think about this, but we don't have that luxury. So I will say yes, because all other things being equal, had we gone back in time and had Alice not drank the potion and observed what happened, we would know that she would not have shrunk. And so therefore the substance, the potion, caused her size change. So counterfactual theory defines relationships between exposures or treatments or interventions and outcomes as causal when the potential outcomes under the treatment are not equal. And so what we mean by potential outcome can be written as Y with a superscript or a subscript. Very often it's a superscript. So here, Y superscript big A equals little a, which is interpreted as the value of Y when the treatment takes the value little a. And so that can represent all of the possible values of treatment. So when we have this inequality that the potential outcome when the treatment is equal to little a versus the potential outcome when the treatment is not equal to little a are different, then this is interpreted again to mean that there's a causal effect of treatment A on outcome Y. And this is the fundamental problem of causal inference, right? We can only observe one potential outcome for any individual, which is the one corresponding to their observed treatment. And we're really just assuming that that observed outcome corresponds to their potential outcome under that same treatment using the SETBA assumption. We cannot evaluate the potential outcome inequality for any individual, again, because we can only observe one at most. So G methods are based on counterfactual theory, and these rely on very strong identifiability assumptions. And so the three that are not italicized are what we call the basic identifiability conditions for counterfactual theory, causal inference, and those are consistency, which refers to the idea that the observed outcome under the same treatment is this potential outcome for that treatment. Exchangeability is the independence of those potential outcomes with the observed treatment, like we would have in a randomized trial. So the probability of A in a randomized trial is fixed, and therefore it is independent of the probability of the outcome. That is exchangeability, the potential outcome. We have to have positive probabilities of treatment. So it must be possible for everyone in the data to be treated with every value of A. And we also assume, but don't really talk about all that often, that we have no model misspecification and no measurement error. So we can't compute individual causal effects, but because of algebra, we can compute average causal effects. And so when identifiability conditions hold, as I discussed on the previous slide, we can estimate the difference in the potential outcomes, which says if everyone was treated and if everyone at the same time was untreated, if there was a difference in those outcomes, then there would be a cause due to the treatment. And under the identifiability conditions, we assume that we can estimate this causal estimate using observed data. We hope in observational studies that we can achieve what's called conditional exchangeability. So obviously, unlike in a randomized study, treatment probabilities are not fixed in observational data, and there's all kinds of individual characteristics and environmental factors that determine what our exposures are. And so our hope is that we can determine all of the variables that are important to control for that are related both to the potential outcome and to the treatment. And then therefore, we will be able to achieve conditional exchangeability. So basically, just like on the slide before, except now, in addition to the treatment, we're going to condition on this set L, which we will think more about. So inverse probability of treatment weighting addresses those unequal probabilities of treatment in observational studies. Observations are weighted by their conditioning on L, probability of receiving treatment A equals little a, so their actual treatment received. Unlike prediction modeling though, the goal here is not to find the model with the smallest residual. The goal is to identify the set L. So IPT, inverse probability of treatment weights, observations are weighted by the inverse of the conditional probability of their observed treatment. There's a formula. Stabilized weights are recommended, and that is because if we use the unstabilized weights, we essentially double the size of our original study population. So what we're doing when we use inverse probability weighting is creating what's called a pseudo population. So in the pseudo population with unstabilized weights, every person is exposed and every person is unexposed on average. And thus, if the potential outcomes under treatment are independent of the observed treatment and L, then the association can be interpreted as causal, given the other identifiability conditions hold. Stabilization ensures that the pseudo population is roughly the same size as the original population, and it helps prevent extreme values in the weights. So selection bias, in addition to confounding, which is really what IPTW is addressing, observational studies are plagued by selection biases, as we know. And structurally, in causal inference, selection bias occurs when we condition on a common effect of two variables, one of which is the treatment or is associated with the treatment, as it is a cause of the treatment, and one of which is the outcome or is a cause of the outcome. Here's a very oversimplified directed acyclic graph, a DAG, which shows us if we were interested in Brock stage as a cause of dementia, which we believe that it is, and we wanted to estimate that association, because we can only measure Brock stage when people have died, we by definition have to condition on this common effect of the exposure and the outcome. So we have to assume that we have induced some form of selection bias by doing this. And this is really, really an oversimplified figure. And this is also a really oversimplified figure, but it adds in some more of the complexity. Because when we're doing these cohort studies and we're thinking about longitudinal cognitive profiles and status, what we're thinking is that really at some time t, right, we have a Brock stage. Sometime after that, we might develop dementia, and then sometime after that, we will die. And then, so what we're observing is the Brock stage at the time of death, which we get through the autopsy. But basically, there's all these different causes of these different nodes, and we also have to think about the selection step that occurs when our participants come to the Alzheimer's centers in the first place. So we have a rather complex issue here that has to be thought carefully about. So with IPCW, inverse probability of censoring weighting, our goal is either to create a pseudo population that represents the entire uncensored population before censoring, or to create a pseudo population that is the same size as the uncensored or complete case population, but without selection bias, and this is stabilized IPCW. So we can have the loss to follow up, those people who are not autopsied, but what we can do is try to make it so that autopsy occurs at random in the pseudo population rather than being associated with very certain kinds of people. So often when we're doing observational research, the causal estimand that we're really interested in is the causal effect when no one is censored. And so this is being explicitly noted here as a C equals 0. So what's the difference when everyone is treated and no one is censored versus everyone is untreated and no one is censored? That's the causal estimate. And so again, with the observed data, we hope that when we have the identifiability conditions and they have been determined to be reasonable, then we can interpret this estimate as the causal estimate. In our case, in this field, we want to know, again, what the association between Brock stage and dementia would be in a world where either everyone is autopsied or autopsy occurs at random. And it turns out that we can use joint weights for confounding and selection bias by computing treatment weights and censoring weights and taking their product. So IPW is a really powerful analytic tool for addressing two of the major threats to validity in observational data. But with great power comes great responsibility, right? So we have to make sure that we are thinking carefully and making it clear what we're doing when we use these methods. So IPW is only valid if the assumptions hold. That's a really key fact. So we have to assess these identifiability conditions or assumptions. And it's difficult to do so, frankly. So the consistency assumption, which is again that the observed treatments and the outcome that goes with that is equal to the potential outcome that we would have observed with that treatment, is outside the data. There's no test we can do, there's no tables we can make. So this has to be based on expert knowledge and study design. Accessing exchangeability, we can use those directed acyclic graphs to do that. The deseparation algorithm will help us. But we are also obligated to examine the distribution of the weights when we do these weighting models. And we want to look at the weighted data as well and examine the balance that we get in these weighted populations to make sure that it's doing what we want in terms of conditional exchangeability. Positivity is a little bit easier. We can do that in the data. Are the different covariates and treatment levels, are they all represented? And we also do this with expert knowledge. So inverse probability of autopsy weights first appeared in the literature about 2009 from a group of investigators from the Adult Changes in Thought study. And since then, there have been at least 8 published papers, usually using the ACT or NACC data that have used these methods. And so we tracked these down and took a look at them, and Taylor tabulated it all up. And so we found that the papers that have applied IPAW to date have not provided explicit evaluation of the identifiability conditions. And as Dr. Nelson would say, I'm not here to gore anybody's ox or name and shame anybody. Just if we're going to use these methods, we need to use them by assessing the conditions that underpin their use. So often in the literature, the weighting model, the censoring model is specified, but there's no justification for the model provided. The distribution of weights is not examined. The weighting model results are provided, like the odds ratios from that logistic model, which is not really relevant. That doesn't tell us what we need to know about that weighting model. And the nature of the weights, whether we're using stabilized or unstabilized weights, is not mentioned, and that's important for readers to know. So if we as researchers or peer reviewers come across IPAW, we want to make sure that we are specifying the variables in the weighting model, which is typically done, and also provide the rationale. State whether the weights are stabilized, and again, provide the rationale. Don't report measures of association from the weighting model. Evaluate the identifiability conditions for IPW, and evaluate the distribution of the weights. So stabilized IPAW weights should be around 1 on average. So to sum up, the nature of clinical pathologic studies means that we must condition on a common effect of the exposure and the outcome, which induces selection bias. IPAW is an appealing tool to mitigate this bias, but it is based on very strong assumptions that must be evaluated each time it's used. And careful assessment and reporting of these assumptions will increase the rigor of our research, and I think also make it more clear what we're assuming when we use them. And thank you very much. I will stop the share.
Thank you very much, Erin. And now we'll hear from Alden Gross, and then we'll move on to the panel.
Hi, everybody. So I'm sharing my screen now. I assume, can everyone hear me?
Yes.
Thank you. So I'm going to talk about an issue that should be readily knowable. In other words, I should have known this before I did it, but I did not know about it. And so I hope that this can be useful to other people. So I'm going to talk about MCI classification in the LASI-DAD study, which is the longitudinal aging study in India. By way of background, there's a great deal of interest in comparing prevalence of cognitive impairment across countries, cross-nationally. The Health and Retirement Study in the United States leads a family of international partner studies. And there are HRS partner studies in, I think, 20 different countries that are partly funded by our NIH. Not all of these partner studies have deep cognitive phenotyping. A couple of years ago, there was an initiative called the Harmonized Cognitive Assessment Protocol, HCAPS, and these projects were designed to facilitate collaborative efforts aimed at cross-national comparisons of cognitive functioning and cognitive impairment, trying to fill that gap. To date, HCAP surveys have been completed in India, Mexico, England, China, EU, and South Africa. I'm going to focus today on data from India's HCAP, which is the LASI-DAD study. It's about 4,000 people and is a subset of the Indian LASI study, which is about 70,000. My goal here is to establish an algorithmic criteria for MCI based on available information in LASI-DAD. And so we're going to apply an approach based on comprehensive neuropsychological criteria using robust norms. And I'm following an approach that was developed for the MexCog project, which is the HCAP in Mexico. There's a great deal of interest in trying to develop an algorithm across these HCAPs that can then be extended to other HCAPs so that we can really start developing common diagnostic algorithms. In other work, we have studied also several dozen adaptations of Peterson's original and revised criteria in LASI-DAD. Some of those are promising. It's not the focus of this particular talk, but I'd be happy to talk about that at another time. Our interest is in establishing algorithmic criteria for MCI. We actually tried clinical adjudications in the LASI-DAD sample, and that did not work well for MCI. It worked very well in this process, clinicians rated LASI-DAD participants using a clinical dementia rating, using an online website. So the clinicians had access to all of the information about function, everyday functioning and cognition, as well as informant reports that were collected in the LASI-DAD sample. And the dementia assignments correspond well with in-person clinical assessments. And these CDRs of one or two or three yield a dementia prevalence of about 7%, which is reasonable. But we found in looking at these data, the clinically adjudicated CDRs yielded about 62% of the sample had a CDR of 0.5. And we didn't really believe that. And there are a number of reasons why there are challenges to online diagnosis, particularly for the MCI phase. You know, there's clinicians, and this is information from the clinicians doing the adjudications. There was insufficient information with respect to the nature and severity of various physical disabilities that might explain some problems. There was not a lot of information about underlying medical conditions and how they might be affecting cognition. There was also a great deal of inconsistent information. So a person may perform well on a brief memory test, but they report subjective difficulties, or the participant and the informant provides contradictory information. There's also a serious challenge with respect to illiteracy. About 50% of the LASI-DAD sample is illiterate, and this is also true of HCAP samples in South Africa and Mexico. So this is an ongoing challenging reality. There are situations in which memory is intact, but other domains are impaired, and there are potentially language issues and issues with the depression scale. So this is the information that we have available in the LASI-DAD protocol. There were a number of cognitive tests, and these cognitive tests were designed first for the HCAP in the United States, which was, they collected data on a subset of HRS participants. So there's a robust battery of tests, many brief assessments from that you would see in mental status exams, but also a CIRAD wordless learning task. There was a symbol cancellation, as well as logical memory, and a couple other tasks, including a go, no go, that turned out well in this sample. There was also a number of informant interview forms, such as the JORM IQ code and the CSID, the Cognitive Activities questionnaire, as well as a 1066 brief informant questionnaire. So this is the information that we have to work with. With respect to the cognitive data, in previous work, we had evaluated the factor structure of cognition in LASI-DAD, and the goal here was to demonstrate, the goal of this paper was to demonstrate that that a hierarchical confirmatory factor analysis composed of general cognition as well as broad domains and narrow domains of cognitive functioning fit the data in LASI-DAD really well. And this model was also happens to fit the data really well in the HRS's HCAP study in the United States. So at least we can be talking about the same constructs. So memory is memory in India, and it's also memory in the United States and in other settings too. So that's great. We have these hierarchical or these broad and general domains, and we wrote in the paper that these broad domain factors may be used in future research to rank individuals with respect to cognitive performance and to classify cognitive impairment. And so that was our next step. So I'm going to jump to the second bullet here. We endeavored to use do a comprehensive neuropsychological approach using robust norms, and we're following procedures set forth by McDougal researchers. In this procedure, we first identified a robust normative group, and in this robust normative group, we're hoping to identify individuals who have no evidence of cognitive impairment. So they have no self-reported cognitive impairment, no history of stroke. They have ideally they should have a low depressive symptom count, and no evidence of functional deterioration. Within this robust sample, which ended up being about 10% of the LASI-DAD sample, we regressed domain-specific cognitive factors that came from this hierarchical confirmatory factor analysis. And so domain-specific cognitive factors, we're looking at orientation, memory, executive functioning, language, and visual spatial ability. We regressed these factors separately on age, sex, and years of education within the LASI-DAD sample. We estimated residuals. And so we then defined domain-specific mild cognitive impairment as performing at 1.5 standard deviations below the mean of any domain-specific residual. So that's what we did. This work is, to a degree, a work in progress, and I just wanted to highlight that specifically identification of the normative group is an ongoing challenge. Certain things are easy to implement, such as, we can easily ask participants if there's a history of stroke. But one of the things that I mentioned is we're interested in identifying a subgroup that has a low depressive symptom count. This ended up being surprisingly difficult. And so what I'm showing to the right here is a histogram of CESD 30 scores. And one would typically expect a highly right-skewed distribution that our previous speaker would love to apply a negative binomial distribution to. We get a distressingly normal distribution in which mean depressive scores are the mean number of CESD score on a CESD 30 is above 10, when typically we use 10 or greater as the cutoff for this. So we think that there are translation issues with some of these items. Some of these items in the traditional Western CESD are not measuring depressive symptomatology in the Indian population. Informant and self-reported functional declines were not asked consistently, and impairment based on the CSID or the JORM IQ code is complicated. So I just wanted to highlight those things. But what I really want to focus this talk on, once we have our normative group, we regressed our domain-specific cognitive factors using these broad domain factor scores. And the idea was we'll just use these factor scores directly from that published hierarchical CFA, because it's a great paper, right? These are the results that we got. and we were able to compare these prevalences directly with the MEXCOG sample, which is the HCAP in Mexico. And so domain-specific prevalences of MCI are rather similar across contexts, and we don't have a hypothesis for whether these prevalences would be higher or lower in India versus Mexico, but I would have a priori assumed that on a population level, prevalences would be somewhat similar. However, the overall prevalence of any MCI is a third of in India of what it is in Mexico. So we were wondering, how could this happen? And through some exploratory data analysis, we realized that all of these factor scores that we're using from this hierarchical model are all correlated at 0.9 or higher. So then I wanted to look back at the hierarchical CFA and its assumptions. So as a primer, this is an example of a single common factor model for a specific domain, such as memory. And these latent variables in circles are assumed to represent the common covariation among observed indicators, cognitive test items that are asked of people. And these are in boxes here. So this memory latent variable represents the common covariation. We could estimate a single factor model for a particular domain, or at right for general cognitive performance. The latter does not give us domain scores. We would only get a single latent variable for general cognitive performance. So we can augment this second model with narrow domains. And this is the hierarchical model that we developed and thought we should use. And this hierarchical model is nested within the common factor model. However, latent variables for narrow domains, such as memory, executive, language, or visuospatial, are not the same as those you would get from a single domain factor analysis, unless you were to force the residual variances of these narrow factors to be 0. In other words, assume that these narrow cognitive domains were measured with no error, and you would have to force the loadings on the broad general factor to the narrow domains to be 1, indicating a perfect relationship. That is to say, if we assume that the loadings on the broad general cognition factor to each narrow factor was 1, and that there is no residual error or no residual variances for the narrow factors, and furthermore that the loadings and thresholds of indicators are the same as a domain-specific single common factor models, then factor scores from that hierarchical model would be the same as from a single domain factor analysis. We typically would not impose such restrictions. To go to such lengths would really return us to a domain-specific single common factor model. So, then we can ask ourselves, why don't we just use factor scores from the single factor model? So separate factor analyses for orientation, memory, executive functioning, language, and visuospatial functioning. Once we do that, then results start to make a little more sense. And so I'm showing the same table of prevalences of MCI subtype by country as I showed earlier, except I'm showing a new set of numbers in the middle. And these subtype or subdomain specific mild cognitive impairments are all similar to each other, and they're relatively unaffected by switching which factor score we use, except the overall prevalence of any type of MCI jumps from 12.5% to 22%. And this is happening because these factor scores are less correlated with each other. That means that if, say, an individual is flagged as being 1.5 standard deviations below mean residual on the memory score, then if the memory score is less correlated with, say, the executive score, then that individual that's flagged as having MCI due to memory has less risk of also being flagged as having MCI due to an executive subtype. The prevalence is still lower in the LASI-DAD sample than in MEXCOG, and MEXCOG used a similar approach to single domain factor scores. They used Z-scores of individual domain-specific tests. So it does appear that the prevalence is a little bit lower. But again, we're still working on issues around our normative group. So our main conclusions are that hierarchical factor analysis is really useful for describing the factor structure of a cognitive test battery. And we used hierarchical factor analysis methods to evaluate whether the factor structure of cognition in the LASI-DAD conforms to a Telhorn-Carroll CHC theory of human cognitive abilities. That has values, but when it comes to empirical estimation of factor scores that one will then use, we suggest using domain-specific single factor models to estimate those scores. There's still a lot of work needed to be done in really developing cross-nationally comparative algorithms to compare MCI, but we're making progress. Ultimately, any approach is going to have advantages and disadvantages, and thus our overall strategy will be to evaluate a variety of methods using data for criterion validation from future study waves that have not yet been collected. And so, thank you. I stop sharing my screen.
Thank you, Alden. We have slightly more than 10 minutes of discussion. I would like to share some thoughts initially. I'll go very quickly. Both presentations were great, and they actually have some common theme that I would like to discuss and open up for further discussion. The first paper by Erin, the first presentation, she brought up some issues of inverse probability weights and how to address the causal inference problems. And there was a call for careful assessment and reporting of analysis assumptions that I found really useful for us in the data course. The second presentation by Alden, who talked about data harmonization for of two studies, was also, I took it as a call for developing cross-nationally comparable algorithms for MCI, but I take it for all kinds of diagnostic groups. And I know, Alden, you have lots of experience for harmonization, for data harmonization in general. Both these actually fit very well in some of the ADRC panel recommendations that how we should work both within NACC, but also outside the ADC network, how we can optimize the utilization of our resources, of our data, but also increase our interactions with non-NACC data. And this is actually what Alden talked. And the interesting part about Erin's presentation was the selection bias. And we know that all our centers have different research interests, which means that we have different selection mechanisms. And if we ignore these mechanisms, then we have bias. Erin's talk was on clinical pathological data, but we have similar selection bias on the longitudinal data analysis. On combining data sets from different studies, we really need to think carefully about the outcome harmonization and the diagnostic outcome harmonization that Alden will be really helpful on that. And really the lessons I take from these presentations is, I think there is a responsibility for the data course for each center to map the selection mechanism within each center and integrate into the statistical analysis that we do, but also start thinking of how we integrate the analysis from different data centers within NACC, which have a diverse selection mechanism. So I would like to ask Erin about that. And on the data harmonization with use of non-NACC data source, I would like to ask Alden, what are the biggest problems that he has encountered? Thank you.
Thank you, Yorgos. That was a great summary. As far as each center, like if we're looking at multi-center data sets, I think it makes the most sense to construct the weights within each center and then consider meta-analytic approaches sometimes on those weighted data sets. You can obviously create weights that take center into account, like center can be one of those predictors of selection. So there's different ways to do it, I think, depending on what the research question is. But yes, it's very important.
So Yorgos, you asked what is the biggest challenge that I've faced in harmonization in this particular, well, I'll talk about within the HCAP projects. I would say the biggest challenge is variability in how tests were administered. We can figure out translation things, and there's a whole suite of challenges around translation of tests. But you know, when you're presenting a CRAD word list, is the CRAD word list just read to participants, or is it read to participants and are they given a cue card with the words? So is there a visual presentation and an oral presentation? Sometimes this is documented. It is not always documented across the sites. And you know, I think it really needs to be, because that can affect one's performance. And so that's a challenge. And this is a challenge not in terms of identifying whether a cognitive test is a good cognitive test or not. We believe that all of these tests are good cognitive tests that represent cognitive functioning. The challenge is finding linkable cognitive tests that are common across contexts. And that's really the big challenge that we have.
Thank you both. I would like to ask Mark, now the second panel discussant, to share his thoughts.
Thank you, Yorgos. I would just express the same sentiment that Dr. Nelson did before. I appreciate the opportunity to come to the data core, and although I do feel a little bit like an interloper, and I will confess about the only part that I understood from Dr. Abner's excellent presentation was the quote from Spider-Man. But I appreciate Dr. Gross's attempts at a cross-national effort to improve upon MCI classification. We've been involved in some of this work for some years, and you know, there are two basic approaches that we've applied. One is this more statistical algorithmic approach to phenotyping folks with MCI. And then another is more of an actuarial decision-making where we attempt to more flexibly examine cognition across various data sets with decision rules. And so we've applied a 2 or more measure within domain impairment that we've defined below 1 standard deviation in terms of demographic norms. And so the two different approaches contrast in the latter being able to apply diagnoses at the individual level. And so I guess I would ask Dr. Gross how some of the methods that you're championing will then translate to diagnoses at the individual level.
So, we have to be careful because we're not trying to say that we're doing a clinical diagnosis. That's why I tend to, especially when I write about this, I tend to try to say that we're trying to find algorithmic classifications for people. These are going to be the most useful use of these scores is ultimately going to be to estimate prevalence across these samples, although there are already folks interested in using the classifications across individuals. I think that can be done with the acknowledgement that there is error in estimation, as always. So.
Absolutely, and that's an excellent point. Do you see some bridging to the use of biomarkers or prediction of progression, or ultimately pathology markers in each of these data sets internationally?
Absolutely. In fact, there's a research group already looking at structural MRI variables across MCI, dementia, and cognitively normal within the LASI-DAD sample, and they're finding interesting differences.
Right. I'll go back to Dr. Malek-Ahmadi's talk earlier in the session that we found dispersion to be useful in predicting entorhinal cortex atrophy over time. So looking at some of these other markers beyond total scores that go into your hierarchical factor analyses, have you looked at variability within domains? Have you looked at process or error scores that come out of some of these tests? So a person may get a total score in the normal range, but maybe it's marred by intrusion errors and things like that.
Yeah, I have not looked at the error scores from the LASI-DAD study, but in other work, I found error scores to be highly skewed. They are, as I'm sure you know, they are quite difficult to work with. There are lots of zeros in these scores. They tend to not fit into latent variable models that well. For that reason, but they do have value, particularly when you're trying to measure things like what we call executive functioning. I think there's value in that.
If I can, just perhaps one more question. I noticed that going into your models a little bit further, you did separate out recall from recognition, which I found to be interesting in the sense that as a neuropsychologist, we make distinctions between recall and recognition. So on the one hand, if you have impairment on both recall and recognition, that's evidence of rapid forgetting, whereas if you have intact recognition but impaired recall, that connotes more of a retrieval difficulty, but the item is still within the memory store, so to speak. Have you noticed any distinctions along those lines within your samples? Because they may relate to the possibility of subcortical ischemic vascular disease in the impaired recall, intact recognition, but more of a neurodegeneration due to Alzheimer's disease and the impairment of both.
So I've actually not looked at sort of these contrast scores between recall and recognition, but that's something that that I'll look into. That could be interesting. Thank you for that idea.
Okay.
And we are at 12, which concludes our meeting. Thank you all for this amazing session. Heather, do you have any further comments before we close?
That I believe the poll questions will be put up. So for folks that want to answer those, please do that. And looking forward to see everyone that can attend the next section in 20 minutes.